touch on that. We've covered some of this when we talked about the coronary vasculature, the coronary system, and we're going to bring these ideas home now today to talk about the blood vessels in the systemic and pulmonary systems. Now, one of the hallmarks of the whole system is that it's highly branched. First, that all of the blood coming out of the right side of the heart comes out of the right side of the heart through the pulmonary trunk or the pulmonary artery, and out of the left side comes out of through the aorta. And then these single tubes then further and further branch and branch and branch to carry blood throughout the system, particularly on the left side. The right side, they'll be carrying blood throughout the pulmonary circuit. On the left side, they'll be carrying blood throughout the systemic circuit. But because they begin with a single tube, and then they branch, the surface area of the walls is going to be increasing and increasing. Now, you remember from our discussion on the coronary arteries that we said that most of the resistance to flow occurs because of friction between the blood and the wall of the tube. So as we go from a single tube coming out of the heart into multiple arteries, then arterioles, and then capillaries, then we'll see that there's this tremendous amount of branching. Because of the branching, there's an increase in surface area, so there's an increase in drag. In other words, an increase in friction. So much of the energy which has been imparted to the blood in the ventricles by the contraction of the myocardium, much of that energy is dissipated through friction between the fluid in the blood and the wall of the tube. In addition to the increase in surface area, the vessel diameter becomes smaller and smaller and smaller. And you remember again from our discussion on the coronary system that as the diameter of the tube gets smaller, there's an ever greater proportion of the fluid that's in contact with the wall, and that's where all the friction occurs. So now not only are we getting a greater surface area because of the branching, because of the smaller and smaller diameters, we're getting greater and greater friction as well. And I'll show you a slide where the energy, the pressures of the blood are dissipated mostly across the arterioles and across the capillaries. But in the end, because so much of this pressure is dissipated through the arterioles and through the capillaries, the branching system of the veins and venules is in fact a low pressure venous return. This is back to the idea we emphasized earlier, that although there's a fair amount of energy imparted to the blood as it leaves the heart, the return system is a low pressure return. And that's because so much of that energy has been dissipated along the walls of the vessels on its circuit through that closed system. The other idea we'll emphasize here is that it's always a closed system. Unless something's broken, it's a closed system, and the blood is always retained within those tubes. And we'll talk about that a little bit. And then we'll talk about blood. And again, there's always this sort of discussion here. Should we go with blood first and then talk about the pumps and the tubes, or do we start with the pumps and the tubes and go to the blood? I like the pumps and tubes first, so we start with that and then we go to blood, but you can do it either way. And what we'll talk about then are the components of blood. And we're going to find that when you talk about what's in blood, there's not all that much there. In other words, about 55% of the blood is plasma. Plasma we're going to find is a protein solution in water. About 45% of the blood is hematocrit, and these are the erythrocytes, sometimes known as the red blood cells. And we'll walk our way through a description of the red blood cells and emphasize they're not true cells. They've lost so many of their cellular components that we call them something called a formed element. And then finally, you'll notice that 55 and 45 add up to 100. Less than 1% of the blood volume is in something known as the buffy coat. And the buffy coat contains leukocytes, which are cells of the immune system, along with platelets. And the platelets are involved with blood clotting. And the platelets are simply these membrane-bound bags of clotting factors. And if you put your phone away, I'd appreciate it. And what we're going to talk about is the composition of the formed elements. The erythrocytes, being about 45% of the blood volume, are the lion's share of the cell types inside the blood. But these really aren't true cells. When we walk our way through them, we're going to find that these erythrocytes have lost their nucleus. They've ejected their nucleus in the process of development. They've gotten rid of their mitochondria, so they're no longer able of supporting oxidative phosphorylation. They've deep-sixed all of their ribosomes and all of their endoplasmic reticulum, so they're not capable of making proteins anymore. These erythrocytes are shadows of their former selves, and they end up being bags of hemoglobin. So that leaves it up to the red bone marrow, where all of the cells of the blood system are produced. It leaves it up to the red bone marrow to be continually making new erythrocytes. So throughout your life, you are generating several tens of millions of erythrocytes every minute of every day. You have to do that because these are not true cells. They don't contain their own protein synthesis machinery, and they will, within 120 days or 90 days, they'll be just no longer useful, so you have to recycle them. So you recycle the components and make new cells. The platelets, we won't spend a whole lot of time on. If you take another course, particularly in hematology, you'll spend a whole lot of time on these, because the platelets are responsible for initiating the process of blood clotting. Now, you may remember that when we talked about blood, we said that it was a connective tissue, and one of the hallmarks of connective tissue is having cells embedded in a protein matrix. And that seems counterintuitive for blood, because it seems to be a liquid. But in fact, that liquid, the plasma particularly, is chock full of proteins, and those proteins are surrounding the formed element. So it comports to this idea of a connective tissue. What's more is we're going to see that it comports this idea of being a connective tissue, and that it's going to be produced in the red bone marrow from hematopoietic stem cells. So you recall that we had those stem cells of the different types of connective tissues, and it's the hematopoietic stem cells which give rise to all of the cells in blood. And we'll walk our way through a couple of examples of this. One of them will be the hematopoietic stem cells that make platelets. These are the megakaryocytes. And the hematopoietic stem cells give rise to megakaryocytes, and the megakaryocytes then send processes, membrane-bound processes, out between the endothelial cells. And those membrane-bound processes then break off in the blood and form these platelets. The platelets are just membrane-bound bags containing vesicles with clotting factors. And the clotting factors then are contained with the vesicles. And if you cut yourself, then those platelets begin to rupture and release those clotting factors. Now, we're not going to spend much time on clotting factors in here. It's this long list of like 32 different proteins, and I will never ask you to memorize that list. But if you do go on, you'll recognize that what this list of clotting factors is determining is a highly regulated cascade of events. And it makes sense that you should have to regulate this cascade of events that generate blood clots. Because if you start generating lumps of blood, which we call blood clots, and they begin to lodge in the fine capillaries of your myocardium, you have a heart attack. If they begin to lodge in the fine capillaries of your brain, then you have a stroke. If they begin to lodge in the fine capillaries of your lung, you now have a pulmonary embolism. So all of these clotting, this inappropriate clotting of blood, leads to very bad things. And so one of the hallmarks of the blood clotting cascade is that it's highly controlled. And when you lose control of it, then you have a very serious medical event that has to be dealt with. So we'll walk our way through all that, and then finally we'll come to plasma and talk about its components. And then if there's time, we'll spend a little bit of time talking about the leukocytes. So what I want to begin here is take this panel and talk about the blood vascular compartment. In the blood vascular compartment are going to be the arteries, arterioles, capillaries, venules, and veins. This is true for both the right and left side of the heart. The arteries lead away from the heart. I really should qualify that. The arteries carry blood away from the heart. Now, coming out of the right side of the heart will be the pulmonary artery, coming out of the left side of the heart will be the aorta, and they then fairly quickly branch into large arteries. But then these large arteries further branch into arterioles that carry blood toward capillaries. The capillaries are the exchange vessels. The capillaries are composed of a single layer of endothelial cells. While it's true that all of these vessels will be lined with a single layer of endothelial cells, when you get out into the capillaries, there's only a very thin, wispy layer of connective tissue, basically basal lamina, around these capillaries. And these are the sites where we have all exchange. This is where oxygen is going to diffuse out of the blood into the tissues, where carbon dioxide is going to diffuse from the tissues back into the blood, and it's the site where nutrients are going to diffuse out into the tissues and waste products back into the blood. So these are the exchange vessels. Then the blood in the capillaries is gathered up into venules, and these then carry blood away from the capillaries. And then the veins carry blood back to the heart. Now I'll try to make some crude drawings here, and we're going to now impose on these vessels a couple of other layers. We're going to see that there's a connective tissue layer, particularly around the arteries and arterioles. But more important than that, there's going to be a layer of smooth muscle. So if we draw this, and we label this as the branching of the arterial vessels. We'll start out here. We can even call this an artery, if we'd like. And the artery then will branch into these smaller vessels, which we'll label here as arterioles. And then these arterioles will further branch into the capillaries. Throughout, there is a continuous layer of these endothelial cells. They'll be continuous along the arteries and the arterioles, all the way out here into the capillaries. But if we take a closer look at a capillary, a capillary is composed of a single layer of endothelial cells. Now, remember that while I'm drawing these as two parallel lines, this is a proper cylinder, so this goes round. These endothelial cells, as we saw with epithelial cells, are oriented, so they sit on a basal lamina. And they have a luminal surface, which is the apical surface. And basically all that's between the blood in the capillary and the tissues underneath is this single layer of endothelial cells. They're very fragile structures. But as we work our way back from the capillaries, we see that we get ever-increasing layers here of connective tissue. This is usually connective tissue proper. But more important than the connective tissue proper are these bands of structures, and I'll just try to draw them as sort of this snake-like structure. And what I'm trying to imply here is that there are these bands of smooth muscle that wrap around the arteries and arterioles. This is intended to represent smooth muscle. And that smooth muscle continues around the arterioles. Now, you recall from our discussion of the autonomic nervous system that the three main targets of the autonomic nervous system are cardiac muscle that we've discussed, and glands we've barely touched on, and smooth muscle. So this is smooth muscle, and this smooth muscle, the smooth muscle, is innervated by the sympathetic branch of the autonomic nervous system. So that these smooth muscle cells contain I put it here, alpha-1 norepinephrine receptors. Now, where there were specific norepinephrine receptors in the heart we called beta-1 receptors, these are now alpha-1 receptors. So they're a different G-protein coupled receptor. The function of these alpha-1 norepinephrine receptors is to trigger smooth muscle contraction. So let's think about this for a moment. Let's imagine that we activated the sympathetic branch of the autonomic nervous system and caused this smooth muscle on this particular arteriole to contract. What's the contraction of this arteriole going to do to the diameter of this blood vessel? It's going to make it smaller. So now remember, Dr. Posseles said that flow is equal to the differential in pressure times pi r to the fourth divided by 8 nu L. There's the character we're interested in. What we're saying is that by reducing the diameter or radius of this arteriole, by basically constricting this and thereby causing R to go down, we're going to reduce flow by a whole lot. It's an exponential. It's an exponential raised to the fourth power. So that means it doesn't take much in the way of a change in the radius of this tube to now shunt blood into these other areas. We see this all the time. And many of you have had this experience. You go outside on a cold day and you don't have your gloves on. And at first your hands turn red. But then after a little while you look down at your hands and they're white. And the reason they're white is that the cold has signaled the autonomic nervous system saying, you need to retain more heat in your core. So we're going to shut down blood supply to the skin. And so you'll start to constrict these vessels and have less blood flow out here into the periphery. So we do this all the time. And what's more is that by regulating these smooth muscles in different parts of the body, we can regulate whether or not we want to send blood out to the skin or send blood into the core and into the gut. So these are important regulatory sites simply through the smooth muscle. And if you start getting down into the weeds of this, there's a whole nervous system out there that's regulating the flow of blood through different parts of the body. But just recognize that by changing the diameter of the tube, we can now change the resistance and thereby change the flow. So any questions to there? Yes? So can blood vessels heal themselves if they're broken? The answer is yes. The answer is if we break these things and then we form a blood clot and we block the flow and basically keep from bleeding out. If we've actually damaged these, there will be signals that will be sent out from the tissue to these endothelial cells and tell them to divide. And they can actually directionally grow back toward places that have been devascularized. There's something called angiogenic factor, and it's also called VEGF. And these are endothelial-derived growth factors. And what they do is they're signals to tell these cells to divide and provide proliferation of these blood vessels. And in some cases, there's a mistake in this, and you get blood vessel proliferation where you don't want it. And some of this happens in the retina of the eye. In the retina of the eye, there's something called macular degeneration, and there are different forms of this. But there's one form of it where you get a proliferation of the blood vessels, and that now begins to cover over the ganglion cells and the photoreceptors so that you can't see very well anymore. And they've begun treating those forms of macular degeneration by injecting antibodies to these growth factors that are responsible for the proliferation of blood vessels. And have had pretty good success with it by blocking the endothelial-derived growth factors and keep them from proliferating the blood vessels in that particular case. So you're absolutely right. There are signals which allow these endothelial cells to proliferate and grow blood vessels back to the old place. And in some cases, this is one of the first treatments for cancers because they were worried about if they could shut off the blood supply to a tumor, perhaps they could in fact affect the survival of the tumor. So some of the early experiments in chemotherapy were to try to control these endothelial-derived growth factors. There's another question. Yes?
So for one of those norepinephrine receptors, if one, like say one of them was stimulated, how much of the smooth muscle would contract?
So the question here is how much of a segment of the blood vessel is going to be covered by smooth muscle? I don't have a good answer to that. These smooth muscles, some smooth muscles are individual cells. A lot of smooth muscles are all connected by gap junctions. These are pretty well connected by gap junctions. I don't know what signals the beginning of a smooth muscle and end of a smooth muscle. That's presumably known, and I just don't know what it is. But you can get these different branches in fairly close proximity to shunt blood one place or another. Anything else? Well, let's look at a couple of slides. The first slide is just one we've seen before and emphasizes the closed nature of the cardiovascular system. Again, emphasizing the idea of carrying blood away from the heart, whether it's deoxygenated blood toward the lungs or oxygenated blood toward the systemic vasculature, always coming out of either the right or left sides of the heart into a single tube, the aorta or the pulmonary artery. And then these branch into arterioles, whether it's in the pulmonary capillary beds or it's in the systemic capillary beds, these arteries will branch into arterioles and then finally into these very fine structures, the capillaries. In some cases, these capillaries are so small that they're only carrying a single erythrocyte one at a time. The erythrocytes are marching through these capillaries in single file to give you some idea of how tiny these capillaries are. And then the blood is gathered up into the venules, back into the veins, and sent back to the heart. And so the pump now of the heart is imparting energy to the blood to move it through the vasculature. And that's shown here. And we've seen this before. We use this to describe the pressures generated in the aorta to move the blood through the coronary vasculature. Now what we're seeing here is measurements of the pulsatile arrangement of the beating heart. So we go through systole, diastole, generating these pressures out in the aorta. And as we get farther away from the heart, we see that this pulsatile arrangement, the pulsatile beats of the heart, begins to smooth out. So that by the time they get out into the arterioles, we no longer see the pulsatile movement of the blood. So, but the main idea here is that there's a dissipation of energy in the form of a drop in pressure as the blood moves through this system. Because remember, as we go from the aorta to the arteries to the arterioles, these blood vessels are branching and branching and branching. And as they do so, the surface area against which the blood is being pushed by the cardiomyocytes is getting greater and greater. And as that surface area increases, the friction of the fluid, the blood, along the walls increases, so that energy is being dissipated as we move through this system, and we recognize the dissipation of that energy as a reduction in the pressure. If we follow this through, we see that most of the reduction in the pressure occurs in these arterioles, the final branches, and then a fair amount too across the capillaries. Tremendous surface area of the capillary beds. So we have the dissipation of the pressure, and now by the time we go to the venules and the small veins and the large veins and back to the heart, we're now looking at a low pressure return. Now, what you have to hold on to here, and it's a little counterintuitive, is that even though the pressure is being dissipated and even though the pressure is going down, there still has to be the same amount of flow. Because if we go back to the previous figure and we look at this figure, because it's a closed system, when the left side of the heart pumps and moves blood out into the systemic vasculature, that same volume must be pumped from the right side of the heart out into the pulmonary vasculature. And when the blood, deoxygenated blood, comes back from the systemic vasculature into the right side, or the oxygenated blood comes back into the left side from the pulmonary vasculature, that volume coming back has to be the same. You can't end up with a mismatch where you're pumping more out one side than you're bringing back into the other. You'll run out of blood because these two pumps are laid out in series. So it's set up in such a way that you have to balance the fluid moving out the two sides. So hold on to the idea of the dissipation of the energy across the arterioles and the capillaries, and then with this figure, simply recognize the degree of branching. And what they're trying to show with this figure is that these are all the way out to first order arterioles. You could follow this back through many arteries. But these are shown with all this branching all the way down to the capillaries, and then the blood is being collected on the venous side and heading back to the right side of the heart, if it's the systemic vasculature. And so the main idea here is out of the capillaries. These capillaries are only a single cell thick. That's where all the diffusion occurs. So remember, all of the movement of oxygen and carbon dioxide, all of that's simply done by walking down a concentration gradient. It's all done simply by diffusion. So any questions up to that? That pretty much brings us to the end of the cardiovascular story. And now we can start on the blood. Now, again, as I said in the introduction, whether you run through these discussions starting with blood, describing the fluid moving through the system, or whether you start with the pumps and the tubes, is pretty arbitrary. But now what we're going to do is talk about the components of blood. And we can think about the components of blood from pretty much an ordinary everyday event. And all of us have gone into the doctor's office, and the phlebotomist comes out with a needle and a tube. And they tie off your arm with a rubber tubing, and then they find a needle and they insert that needle into the side, and then the low pressure return fills the tube with blood. And then, although it used to be done right there in the doctor's office, now they take it off to a laboratory. They send that tube off to a centrifuge. So this tube of blood, and I should say that these tubes are usually lined with some sort of anticoagulant, usually heparin, so you don't get blood clots. But then they take this tube of blood and they put it in a centrifuge, and they spin it in a centrifuge. When the blood is spun in a centrifuge, there are downward forces that are imparted onto the blood. And those downward forces are such that the denser materials will move to the bottom of the tube. And when we do that, we get a distribution that looks a little like this. There's the tube. And then at the bottom of the tube, there are these erythrocytes. Sometimes we refer to these as a red blood cell. These erythrocytes aren't true cells, and we'll emphasize that in a little bit. They're really just bags of hemoglobin, but they compose something in the neighborhood of around 45% of the blood volume. This is called the hematocrit. And sometimes you'll see the hematocrit referred to on a blood panel. And it will give a number like 42 or 45, and that means that 42 or 45% of the blood volume is these erythrocytes. Then we find that in this upper portion, up here, this is the plasma. And if we break down what's in the plasma, we find in the plasma there's 90% water, something like 8% protein, and something in the neighborhood of about 2% other. And when we look at the overall percentage of the volume that's plasma, we find, taken together, this is about 55% of the blood volume. So effectively, almost all of our blood is made up of this plasma, which is basically a protein solution, and the underlying structures here, the erythrocytes. Now I should add here that the reason the erythrocytes are being moved to the bottom of the tube is that the hemoglobin contains iron. Iron's a pretty dense element. There's so much iron in the hemoglobin that it increases the density of these erythrocytes, and because of the high density, they move to the bottom of the tube. Then there's a very narrow band in here. It's a very narrow band that resides just above the hematocrit and below the plasma. And this has a name called the buffy coat. And the buffy coat comprises something like less than 1% of the blood volume. The buffy coat contains leukocytes. This comes out of the Latin leuco, meaning white, so these are the white cells. These leukocytes we know now are part of cells of the immune system. Another component of the buffy coat are something called platelets. The platelets we refer to as a formed element. And we use this euphemism when talking about blood to describe a formed element when we mean it's not a true cell. where the leukocytes are true cells. They are full-fledged cells. They contain a nucleus, they contain mitochondria, they contain ribosomes and rough endoplasmic reticulum, the whole 9 yards. These are true cells. Platelets are in fact a cell fragment containing clotting factors. In vesicles. And as we walk through this, we're going to emphasize, too, that the erythrocytes are more properly termed formed elements. They're not true cells because they lack protein synthesis machinery. So all of the cells, whether they're the leukocytes, whether they're the formed elements of the platelets, or whether they're the formed elements of the erythrocytes, all of those cells originate from hematopoietic stem cells in the bone marrow, the red bone marrow. You may remember from our discussion earlier of connective tissue, when we brought up the idea of blood, we said that the cell type that gives rise to the connective tissue of blood is the hematopoietic stem cell. And those hematopoietic stem cells, at least in adults, are relegated, they're located in the red bone marrow. So what we'll do here now is try to imagine some of these cell types in the red bone marrow and how they're in fact being generated. The first type we're going to talk about is something called a megakaryocyte. Let's take this panel. We're going to describe this as a megakaryocyte. A megakaryocyte in the red marrow. Now, the red marrow, like all other tissues, is well vascularized, and there are these capillaries in the red marrow. So this is meant to be a capillary in the red marrow. And there will be a cell up here. Put the cell in purple. There's a cell up here, and I'm going to give it a name. It's called a megakaryocyte. The megakaryocyte is often multinucleate. It arises from a hematopoietic stem cell. The hematopoietic stem cell differentiates into a megakaryocyte, and the megakaryocyte basically sits in the red bone marrow, but it just doesn't sit there. It sends out these extensions of cytoplasm surrounded by plasma membrane. And it extends these extensions of its cytoplasm surrounded by a plasma membrane down in between the endothelial cells of the red bone marrow. Then once inside the blood vascular compartment, those extensions of the megakaryocyte break off. And they now are these structures which we call platelets. The function of the megakaryocyte back here was to fill the cytoplasm, and hence the platelets, with vesicles containing clotting factors. These platelets are essential for controlling blood clotting. And in fact, if you talk to intensive care physicians often run into this problem called disseminated intravascular coagulation. What often happens with disseminated intravascular coagulation is that the individual, the patient, has for some reason or another taken up a massive bacterial infection. And they've gotten a blood-borne infection, which we commonly call sepsis. And though the sepsis bacteria, for reasons I don't understand, but the sepsis bacteria have these exotoxins, which now cause the blood clotting factors to generate blood clots. So as a result of some forms of sepsis, these platelets are dysregulated and they begin to generate blood clots. But normally the blood clots are formed outside the blood vascular compartment. But in this case, they're intravenous. So this is what's termed disseminated intravascular coagulation. If you talk to these physicians, this is a very difficult condition to treat, because what happens is in the patient there are all these emboli. The emboli is spread perhaps to the myocardium, and there's effectively a heart attack. To the brain, effectively there's a stroke. To the lungs, there's effectively a pulmonary embolism. And so, if the person's going to survive, you have to do something to treat them for all these emboli. So the first thought is, well, we'll give them a drug that breaks up the clots, so-called blood thinning agents, warfarin or streptokinase or one of the others. And that's true, it will break up the blood clots and return blood flow to those areas that have been occluded by the blood clots. But now you have a new problem, because in the process of making all those clots, the platelets and the blood clotting factors have all been used up. It's now the individual whose first crisis was all these blood clots now has a new crisis with not enough clotting factors at all. So if now you treat them by breaking up the blood clots, and you risk the sequelae of some sort of hemorrhagic stroke or hemorrhagic bleed. So they're always on the horns of this dilemma: which one do you treat, and in which order do you treat them? And it's a really difficult problem, and it's one of the things that you find these critical care physicians most vexed by trying to sort that out. But again, that's just a story to tell you that we're not going to spend a whole lot of time on blood clotting. But if it gets out of control, you're in serious shape. So you really, if you go on and study hematology, you'll have to know all of those series of blood clotting agents, and where they're affected and where they're changed. But for our purposes, just know blood does clot. And if it's out of control, it's a bad thing. But then there's another hematopoietic stem cell up here. I'll put it in red. In the red bone marrow, there will be another cell, which has a nucleus. I'm going to label this as an hematopoietic stem cell. Now, theoretically, if it's a true hematopoietic stem cell, it's so undifferentiated that it could become a megakaryocyte, it could become an erythrocyte, it could become any one of the leukocytes. It could become a T lymphocyte or a B lymphocyte, it could become a macrophage, because it's pretty much totipotent as far as the blood lineage is concerned. But the one we're going to talk about is the hematopoietic stem cell that's going to be urged by a series of growth factors to become a cell type called a pro-erythrocyte. This is basically an undifferentiated erythrocyte. The pro-erythrocyte has all of the normal components of a cell. It has a nucleus, It contains ribosomes. It contains endoplasmic reticulum. And it even includes a pretty robust Golgi apparatus. Now, you'll recognize the ribosomes, the ER, and the Golgi apparatus, and the nucleus as all being required components for protein synthesis. So shortly after this proerythrocyte has made a whole bunch of proteins and it's generated particularly a lot of hemoglobin. Shortly after that, this cell undergoes a change and becomes a new cell type called a normoblast. The remarkable thing that the normoblast does is that it ejects the nucleus and it ejects mitochondria. Now, there are a couple of steps in between the proerythrocyte and the normoblast, and if you take a course in hematology, you'll learn all about those. All I want you to know is we start out with an hematopoietic stem cell, it becomes a proerythrocyte, and that's destined to become a normoblast that gets rid of its nucleus. But recognize how important that is. If you've gotten rid of your nucleus, you're making no more new proteins. So this is a dead-end cell. It cannot survive on its own because it can't make its own proteins. And it will further mature into a cell type here I'm going to call a reticulocyte. The reticulocyte gets its name because under the right stains we can see these remnants of endoplasmic reticulum. The reticulocyte is found in the red bone marrow, but its major importance to us clinically is that the reticulocyte, among all of these lineage of cells, is the first one to go into the blood vascular compartment. So that if we were to think about it back here alongside with the megakaryocyte, this cell will have matured into something which looks like an erythrocyte, but is a reticulocyte. And you can think of these reticulocytes as adolescent erythrocytes because they're going to leave the red bone marrow, they're going to leave home, and they're going to go out into the blood vascular compartment, where they will be, again, called reticulocytes. So the importance of the reticulocytes clinically is that hematologists can recognize them in a blood smear. So they can take a blood sample and then look at the blood sample in the microscope and begin counting the number of reticulocytes. And there's a kind of standard number for the proportion of these mature erythrocytes out here that will be reticulocytes. Because over time, the reticulocytes lose the ER fragments. So these reticulocytes now become a kind of signpost. And since they are juvenile erythrocytes, by looking at the number or proportion of erythrocytes that are reticulocytes, you can get an idea of the rate of turnover of the erythrocytes. And what hematologists are looking for is that they get reticulocyte counts above a certain number, they begin to suspect some kind of hemorrhagic problem. This is especially true in the elderly or anyone who's taking non-steroidal anti-inflammatories where they've affected the lining of the stomach and they generate these small bleeds and they slowly hemorrhage. And the hematologists will look for the reticulocytes to give them a hint that there is a problem in these hemorrhagic problems. With that, I'll let you go, and we'll finish the story next time.